Hello, everyone. Thank you for attending today's webinar, Strategically Accelerate COVID-19 Diagnostics from Development to Market, presented by Precision for Medicine. I'm Rebecca Williamson. I'm the publisher of Fierce Biotech, and I'll be moderating this webinar. In this webinar, our panel of experts is going to discuss strategic approaches that will help accelerate the development of diagnostics for COVID-19, including building a biospecimen foundation for product development, navigating the regulatory landscape for serology tests from EUA to 510K, and leveraging important design considerations for market access. Our speakers today are Karen Richards, Senior Vice President of In Vitro Diagnostics and Quality at Precision for Medicine, Jim Buchel, Vice President of Biospecimen Solutions, also at Precision for Medicine, and David Parker, Senior Vice President of Diagnostic Solutions at Precision for Medicine. You can read their full bios by selecting the Speakers tab on the left side of your window. I have just a few technical notes for you before we begin. The webcast is being streamed through your computer, so there's no dial-in number. For the best audio quality, please make sure that your volume is up. The webinar is being recorded and will be available on demand within 24 hours after the event. And time permitting, we're going to be following the presentations with a Q&A session. So please submit your questions using that Q&A tab on the left side of your screen. All right, I think that's everything from me. David, please go ahead.
Thanks, Rebecca. First, I'd like to lead off with a word about our organization, Precision for Medicine. Precision for Medicine is an industry leader in the design, development, and execution of biomarker-informed clinical research and complex data management. Now, we're applying these capabilities and our deep experience in respiratory and infectious diseases to support the efforts of companies focused on developing COVID-19 therapeutics and diagnostics. Among the services we're providing to the industry, we are conducting clinical trials for COVID-19 therapeutics, consulting on diagnostic and drug regulations, sourcing and supplying critical biospecimen samples, providing BSL-2 laboratory services, including support for immune monitoring, vaccines, antibodies, and all research related to them, and using our biorepository to receive and store every type of COVID-19 specimen. Now today, we'll be addressing three critical challenges for developers of COVID-19 diagnostic tests. First, ensuring that your biospecimens are appropriately sourced and vetted. Second, developing an optimized regulatory plan for the post-EUA environment. And finally, optimizing your test development for the evolving reimbursement environment. So to kick it off, with that introduction, I'll turn it over to my colleague, Jim Buchel, to address biospecimens. Jim?
Thank you, David. I'm really excited to be part of this webinar here today. Over the last 30 years, I've spent my career chaperoning biology to science and providing biomaterials to those scientists so they can translate them into medicines. And during that time, I've gained a profound amount of respect and value for the role of that biospecimen. No matter how you choose to refer to them, as a sample, a specimen, a biospecimen, or tissue, there exist two constants, and they are availability and quality. And given the speed of which COVID science is operating at today, it's become very concerning to me that we are witnessing a separation of quality in an effort to address availability. My intent here today is to share with you some insight and resources that you'll be able to bring into your own biospecimen program to ensure that quality and availability always remain together. So developing a quality diagnostic, it begins with a quality biospecimen. Biospecimens are the fuel to the science engine. They are the source of the discoveries, they are the basis of diagnosis, and they are the informer of treating, monitoring, and response. Now, given the magnitude of the biospecimen, one might assume that there exists a well-defined process from the time the patient is first encountered all the way into the laboratory. But unfortunately, the reality is this is not the case. And while the biospecimen's role remains unchanged, the quality of the research and the resulting diagnostics aren't always the best they can be. Evidence couldn't be any more clear than it has been over the last several months, with COVID-19 assays entering and exiting the market at an unprecedented pace, all under the suspicion of poor quality and inaccurate results. So for the next 10 to 15 minutes we're going to have here today together, I'm going to share with you several key insights and tactical methods that you can deploy within your program that will minimize your time, your resource, your budget, and most importantly, your frustration, and deliver to you high-quality biospecimens for your program. So a great place to start this talk is with a quick look at the traditional resources that you may leverage for COVID-19 patients and specimens. Let's begin with patient registries. There are many of them out there. They can be leveraged for both data and biospecimen research. They are predominantly based within an academic institution or more likely a nonprofit organization. Typically, these types of registries come with limitations of use, so be certain to check first. At Precision, we opted to build our own. We did so in order to provide a quick turnaround within an engaged community, a community that had confirmed their interest in studies, and to return when additional specimens are needed. To most of our clients, the most important benefit of our patient registry is the freedom from limitation of use. And if you happen to be working in a program that is operating under stealth mode, this is a very important consideration. The next source type to consider, that is directed patient recruitment. A good number of attendees here logged into this webinar, they may be affiliated with an academic medical center. And the likelihood is that there are going to be ongoing COVID-19 studies running in these centers. This would be an opportunity for you to potentially piggyback onto those programs to access patient specimens. A little bit further into this webinar, I'm going to share for those of you that are not affiliated with an academic medical center, a direct-to-patient strategy that you can leverage with your own team. The next two resources that I want to speak about, they're often confused, yet they are very different. The first are brokers, and it's likely you've been contacted by many of them recently selling COVID-19 biospecimens. Whether they're individuals reaching out to you directly via email or phone, or they're an online marketplace where you log in and place an order for fulfillment. You have to be mindful. Be mindful of the inherent risks that could impact the quality of your research. Primarily, that risk comes from a lack of standardization. Historically speaking, and absolutely currently with COVID-19, brokers are that one resource where availability trumps quality 100% of the time. And now the last resource I want to touch base on, that is biorepositories. Biorepositories are very similar to biobanks in that they obtain large cohorts of patient specimens and data. However, they differ in that they obtain specimens in a manner that's typical of that of a phased clinical trial, which tend to include 100% standardization and harmonization of the data and the specimens collected, as well as consent for the specimens and data with clear language that there is going to be research involved. The manner in which they undergo these collections are typically done under IRB-approved protocols. And the very last point, and this is real important, is that the biorepositories verify the collection, they verify the data, and they verify the handling and storage of the specimens. Precision is a biorepository, and as indicated earlier, we've been enrolling continuously throughout 2020 in several areas of study. As might be expected, we're heavily involved in COVID-19 programs, and I'd have to say the most valuable program we have ongoing here right now for anyone involved in drug, vaccine, or diagnostic development are our two observational studies. Now, being mindful of the time of those who are online here and interested in just regulatory and commercialization and not biospecimens, I'm not going to go too deep into this at this time. However, if you are involved in COVID research and you'd like more detail on either of these studies, please feel free to reach out to me directly via LinkedIn or on our website. So let's dig into how you would build a valuable biospecimen that's going to fuel your research programs. No surprise here. You have to start with a plan. What I'm sharing on this slide are some key elements pulled from a much more detailed process that we use here at Precision and refer to as a biospecimen plan. You can adopt these processes into your program relatively easily. What I always tend to find very helpful, and I think would work for you, is to start by defining your endpoint first. Then document and figure out your process going backwards. Ask yourself, how are these specimens going to be used? Are you supporting an agency submission? Are you interrogating a biomarker for potential prognostic applications? What will be the data and biospecimen needs for these to answer these types of questions? If you're supporting an internal research program, also known as a sandbox program, the use of specimens with limited data, such as that can be obtained from a remnant specimen, just might suffice. However, if you're going to be supporting an agency submission, then the data and biospecimen requirements are far more complex, and it's suggested that you seek guidance from the FDA online to figure out what that additional information may be. Now, a recent example that we had back in 2019 that I think would be helpful for you to hear about was with an experienced diagnostic company, and they were growing concerned with their study, and it was targeted to start at the end of 2020. They wanted to level check whether their strategy was aligned properly. We set up a meeting to deep dive into the facts and figure out what was going on, and then hopefully come back with the intent of providing some feedback on their program. What was exposed to us during this meeting was that they were using the specimens to support a 510K, yet 80% of the samples are going to be sourced non-US. It has been apparent to us that minimal FDA guidance was not sought initially. The company valued our input. They were a long-term partner of ours, and they urgently asked us to rescue this program. Within a matter of days, we developed a budget and plan. Immediately, we pivoted their program to sourcing specimens using our US-based labs, and thereby drastically reduced any future downstream scrutiny from the FDA. They were able to maintain their program timeline, and at the end of the day, we ended up with a very happy customer, and they got their program done. Bottom line, make sure you talk to all your stakeholders and have clear visibility to the application of these said specimens. So once you've defined your specimen plan, it's very important that you vet your partners. Ask yourself 2 key questions. Does the site or vendor have access to the patient or sample type to fulfill this plan? Do they have the infrastructure necessary to ensure quality and compliance? Some elements to consider here would be, do they have experienced staff that understands what good clinical practice is? Do they have the processing equipment? Do they have a CAP and CLIA certified lab? Do they have the ability to comply with what your programs and reporting asks are going to be? For us, well, we insist that all of our labs are fully vetted to just these minimum standards before we would engage with them. A couple of easy asks of your resources that you might be able to consider. If you're looking at a laboratory, ask if they have sufficient FTEs available. FTEs are labor, okay? Understand that bigger isn't necessarily better. Our best lab providers enroll 2 to 3 times that of the large providers, and they're very small labs. Why is this? Very simple. The program has a much bigger impact to them. So something to ask if you're looking to vet a clinic partner is, do they perform phlebotomy? In most cases, if they don't, we opt not to move forward. However, there are times where it simply isn't feasible to avoid it. A good example of this was a few years back. We were contacted by a small therapeutics firm in Cambridge working in a rare disease. They had relationships with PIs and KOLs who had these rare patients within their practices, but they didn't have the ability to conduct sampling. We offered to help, building a multi-tier program for them that provided both at-home collections or collections that could take place at our biospecimen collection facility here in Massachusetts during a week where that therapeutic company was hosting a support group meeting for these rare disease patients. Ironically, it was the latter that the company opted to lean on most. We enrolled 35 patients, all under the age of 12 in this rare disorder, and we accelerated that program's pipeline by years. One last point before leaving Right Resource, and that is the importance to drug developers, and that is diversity. Diversity in the levels of data, of comorbidities, and geography. All factors that need proper consideration to support downstream analysis as stratification, outcome investigations, and obviously future unplanned research. Bottom line here, if you don't have the right data, you're not going to end up with your quality diagnostic. So just when is the timing right? Partners have priorities. They have CAP inspections. They have JCHO accreditation processes. They even have competing studies. Not to mention partners' priorities change. Nothing more apparent than here today with COVID-19 in the way that office visits and procedures are now going forward. That's going to reduce the patient availability by almost 50%. It's also going to eliminate waiting areas, a common point where researchers can advertise their studies to patients. They're no longer going to be present. And then finally, most healthcare providers are being encouraged to leverage telehealth instead of on-site clinic visits. I won't spend any more time here on this. I simply want to bring it to your attention because if the timing isn't right, well, this is going to impact the specimen availability because there are no patients, and most likely the quality because the site is already thin. So as I touched on the prior slides, in today's healthcare environment, traditional biospecimen plans aren't necessarily going to work. The registries have long applications and review times. The brokers, well, they have questionable supply. COVID-19 wasn't part of their world in 2019. And as we mentioned here, the clinics, well, they have restricted access. So do you give up? No, you definitely don't. In fact, you work creatively with your procurement team in developing a new strategy. We did it, and so can you. With precision support and buy-in from key stakeholders throughout our organization, such as clinical operations, marketing, talk to R&D, and IT, we were able to build a biospecimen strategy specific to be successful in this current environment. Simply put, we ended up shifting the focus from the clinics directly to the patients so that the patients could find us. And we did so leveraging online and social media tactics. We created a presence online, leveraging search engine optimization techniques and keywords, and then we enhanced our participation in social media, utilizing geo-mapping and COVID-specific messaging and hashtags. This strategy has not only worked enormously for COVID-19, but coincidentally, it has led us to new resources and diversity and leveraged the future programs. Now, I'd be remiss if I didn't touch briefly on a key element here that needs to be considered in this type of effort, and that is infrastructure. It's different, and we developed our own with the following considerations. Online recruiting, consenting, and scheduling. How are you going to actually get to the patient? So we've developed a remote clinical team, one that's verifying patient data in the patient's home, obtaining medical release, and obviously performing the collection. And then finally, testing. Where this isn't a standard of care patient specimen, we had to reach out to outsourced labs in order to get the testing done, and therefore had to ensure that they were using EUA-approved methods and had all the right process, licenses and qualifications. The send home here, gang, is if you hit a wall and you encounter an obstacle, don't take drastic high-risk measures, such as loosening your quality standards. Be creative. The possibility and quality of your research depends on it. So finally, developing a detailed biospecimen plan is only going to get you 50% of the way towards a quality biospecimen. The second 50%, well, that's all execution, of which, if your endpoint isn't an agency submission, then it's typically overlooked, or even worse, it's assumed. Yet this is the point that introduces the greatest risk of variability to research, and it typically isn't recognized until long after the testing is complete. You have to have an execution plan in mind. Some things you need to consider are how are your specimens going to be collected, and where are they going to be processed? How are you going to manage the data and the biospecimens? How are you going to keep the data linked to its companion biospecimen? You have to think about how I'm going to scale my program as it grows and as enrollment velocity increases over time. And this includes elements such as supply shipments and staff to receive specimens that are collected. And then finally, you have to understand how the specimens that you're going to receive, are they research ready? What has to happen to these specimens once they come into your facility and need to be reformatted for your researchers? As much as possible, I'd recommend that you focus on standardizing and harmonizing the procedures behind your biospecimen collection and data. This will naturally ensure you're providing your stakeholders 100% quality specimens. So with that, I want to thank you for listening. I hope you found value in some of the content and that we may have provoked some new ideas as to how to ensure you can build a quality biospecimen resource. I'm going to turn this now over to Karen Richards to learn a little bit more about navigating the regulatory landscape.
As Jim has just described, the foundation for developing a robust diagnostic product must include a solid strategy for acquiring samples. I'd like now to discuss some of the current thinking around the development of serology tests for use under the EUA provisions, while thinking ahead for how to maximize the use of samples for establishing performance for the EUA and leveraging the performance towards transition to 510K submission. FDA has updated the current policy for commercial manufacturers of serology tests and provided alternative pathways for marketing these tests. Many of the templates are available on their website to direct applicants on how to pursue marketing of an EUA, including templates for serology and even those for how to think about doing human usability and home collection studies. All pathways described on this slide require that the test be analytically validated, a clinical agreement study be performed, and of course that the product is labeled properly. But each pathway has nuances that are important to point out. On the left-hand side, under the policy pathway, tests can be distributed without FDA review initially, as long as the labeling reflects that the test has not been reviewed by FDA until such time as it has been authorized. And the labeling is limited on how the test can be used. An important factor that's the subject of many of the FDA town hall discussions is that a test that is distributed prior to EUA authorization can only be marketed to high-complexity laboratories. This means limitations on the distribution of the test and is the subject of a lot of conversation during FDA calls. Under the EUA pathway in the center column, the labeling allows for statements that the assays are used as an aid in identifying individuals with an adaptive immune response to SARS-CoV-2, indicating recent or prior infection. Further, in most cases, the authorizations allow for distribution of the test to both moderate and highly complex laboratories, therefore broadening the market and distribution of that test. And in some cases where data is provided to support the application, authorizations will allow for point-of-care use and may even be waived. The third pathway on the right-hand side is the umbrella pathway authorized in April, mainly directed to serology tests based on lateral flow or ELISA technologies. A key factor for the umbrella pathway is that FDA has authorized centralized testing through NCI, where manufacturers can send their tests for independent verification of clinical agreement. This independent clinical agreement can be used in the EUA submission to support the additional data required by the serology template. And FDA recently announced that this NCI panel can now be made available to manufacturers who are developing serology tests. One thing I'll note about any of these pathways is FDA has stated very currently that they are now needing to prioritize EUA reviews only for those tests that require an approval, such as a home collection or a home test. And they're prioritizing this over other tests that can either be distributed through notification or that are similar to other already EUA authorized tests. So as I stated on the previous slide, no matter the pathway to market, the assay must be analytically validated. This slide covers the current state of affairs and key studies required for EUA submission by a manufacturer of a serology test. Starting with class specificity, this is a measure of the test's ability to distinguish IgM from IgG, for example, and ensuring the assay can deliver results for both when both antibodies are claimed. For total antibody assays not claiming to distinguish between IgM and IgG, the class specificity study is not required. Another study is cross-reactivity, and this is the assay's ability to distinguish between antibodies to SARS-CoV-2 versus other viral pathogens. There's a long list suggested in the serology template. But there is a caveat that is long and detailed, but I'm going to tell you a little bit about it here because it's important to consider. So if the 75 samples that you're going to obtain for this serology test, if they're collected in the U.S. prior to December of 2019, and they come from a population that either has a high prevalence of vaccination or had a high infection rate with a particular virus, and you can demonstrate 98% specificity overall with your test, then cross-reactivity testing for those viruses may not be required. So it's really something to think about. And as Jim mentioned, they have access to all kinds of different types of samples. This might be one that you would want to consider talking with him about, is whether or not they have samples from these high vaccination or high prevalence prior populations. However, it should be noted that the umbrella pathway does not specifically require the analytical specificity studies described here. Rather, during recent town hall meetings, FDA stated that if clinical agreement studies are performed using the NCI panel, this, quote, waives, unquote, the requirement for additional cross-reactivity testing if your assay achieves a 95% specificity using the NCI panel. So that's an important thing to consider if you're working with a small budget or you're trying to consider whether or not you're going to be able to do or how to do the other analytical studies for cross-reactivity and class specificity. For any assay using a mix of different sample types, a matrix equivalence study must be performed. This can be done either analytically or during your clinical agreement study and should be performed with the same donor. However, for finger stick samples, unless the stability of collection and transport of sufficient volume can be demonstrated, this is best studied during the clinical agreement study. So that would mean that you would have a serum and a plasma and a finger stick sample collected all at the same time from which you can do these studies. Down the road, we hope that finger stick validations will support the potential for collection and home testing. So we talked about the clinical agreement a little bit. Clinical agreement studies and performance expectations for serology tests are described in this slide. Whether a manufacturer is planning their own prospective study or taking advantage of the umbrella pathway for independent testing, the assay will require a clinical agreement study of at least 30 positives and 75 negative samples. The current information from FDA is that comparison must be performed to an EUA-authorized molecular test, preferably collected at the same time. We hope this changes down the road and that companies are able to demonstrate studies against other EUA-approved serology tests, but that's just not the way things are today. Also, FDA has seen some interference with HIV cross-reactivity in tests, and therefore they're requiring that 10 negatives be tested that are from HIV-positive donors or demonstrate through the cross-reactivity studies that HIV does not interfere with the assay. For assays that are by design simple to use and easy to perform, clinical agreement studies should include multiple operators that can demonstrate use of testing using simple instructions. This will help pave the way for moving these tests closer to other use environments. Serology tests are well suited for home collection and test if validated using a sample type such as finger stick or even saliva. To achieve claims for home collection and test, however, additional flex studies and user studies must be performed that are currently being described in either published or draft FDA templates. So that's the current state of affairs. Now turning from the current to future thinking, how should manufacturers be approaching and preparing to move from an EUA to a 510K? We know that the FDA will require any first test to go through the de novo process, with follow-on using the 510K process. So predicting how this might go forward, we can look at the Zika EUA as a model, going from the EUA to 510K. In this example, where Company A paved the way for entering the EU market early with limited data required, while Company B took an additional year to enter that EUA market. In the example of Company A, the subsequent testing required to support a regulatory submission for FDA review included extensive additional analytical and clinical sample testing, and in the absence of a regulation at that time for Zika, a de novo submission was required. That's in contrast to Company B, where the EUA authorization happened later the following year. Much of the testing performed to support it could be leveraged for the regulatory submission. Because Company A had achieved the de novo, Company B was also able to demonstrate substantial equivalence to Company A rather than to PCR. So they were able to show serology to serology rather than serology to PCR. So there's definitely a balance between testing now for EUA authorization and testing later for regulatory submission. Another consideration might be that the first de novo equivalence, as I mentioned, will require performance against PCR versus a substantial equivalence using the serology. So with the Zika model in mind, an important factor of managing your EUA now is to consider your future submission for 510K to FDA. And part of that strategy is the access to biospecimens, which will be key in determining how to manage analytical studies for the EUA and for future 510K. The analytical studies described above in this presentation can be designed to achieve current state while considering future state, and therefore minimize cost and time to market for the transition. For example, under the current EUA for serology, cross-reactivity can be demonstrated using either natural or contrived specimens. Consider that in comparison to the de novo for Zika, where the use of specimens from patients confirmed to have IgM antibodies to other microorganisms was used to support the approval. So they had to go back and do additional testing to support that. You might consider using natural samples now for your EUA, knowing that you can leverage that same analytical data set for your future 510K submission. So it's always important to strike a balance on cost, sample availability, and study designs to make that transition out of EUA to 510K. So I'd like to say, as you know, there are a lot of players in the market today. Thinking of how to transition your product from EUA to 510K cannot be done soon enough. It may be less important to get in line with your EUA if it means sacrificing study quality, and Jim talked a lot about that before, and it cannot be emphasized enough again here. Well-designed and executed studies for EUA can potentially be leveraged for a 510K submission to FDA later, and that saves time and money long term. As I mentioned to you before, even though Company A got on the market with the EUA a year before Company B, when it came to the 510K clearance, they were only a few months apart. Even though exempted under the EUA, developing your device under design controls now can also save time later when moving the product through its life cycle and into commercial manufacture and distribution. Again, this is very important, especially if you're new to even entering this market. Understanding and getting your product under design controls soon and early is very, very important. Another example, if your EUA is for serum, and that's enough to support the pandemic, but you anticipate being in the market with other sample types, it would be better to collect and store paired samples now in plasma and other matrices, so that can help pave the way for broader claims for a 510K. Early communications with the agency will be so key, and staying on top of the various policies, templates, and methods for authorization will be critical to your success to enter the market now and ready yourself for the future. Be thinking strategically around EUA versus 510K, and map out now how to leverage your EUA studies for future submissions, and what additional studies may be required, such as flex studies for waiver or other user studies for home collection and testing. As regulatory professionals, we're always asking to be brought into the assay development process early, so that we have the opportunity to provide the best strategy to lower the barriers to market entry while providing a high-quality product. And now let me turn the webinar over to David Parker.
Thanks, Karen. In this section, I'd like to talk about some of the key reimbursement factors you should consider as you develop your COVID-19 test, keeping in mind the context and some of the EUA to 510K factors that Karen just mentioned. First, as many of you already know, market access and uptake of a diagnostic test depends on securing adequate reimbursement. In turn, reimbursement is a function of coverage, payment, and the coding that's needed to tie those two factors together. So first, let's talk about coverage. The current coverage environment is good for COVID-19 testing for both Medicare and commercial payers. You might be thinking, as some companies have asked us, I'm planning to launch my COVID-19 test under an EUA. I wonder if I'll have trouble getting covered. Well, the answer is, as of right now, as long as your test is FDA authorized, it doesn't matter whether the authorization came through the emergency use pathway or traditional FDA authorization routes. During the period where demand for testing outstrips test supply, payers are not likely to be any more picky than that. However, and this is important, this situation isn't likely to be permanent. So keep in mind, once FDA cleared tests begin to enter the market and supply of cleared tests can actually meet demand, payers may well become more demanding in their requirements and preferences for coverage. So to Karen's point, the companies that are first to market with FDA cleared tests through the 510K pathway are going to have a considerable coverage advantage in the long run over the laggards that have lingered in the EUA pathway too long. We'll address specifics about coding and payment in the next couple of slides, but I do want to offer a couple of upfront thoughts right now. First, as you develop your test, the key point about payment to keep in mind is to make sure that labs can afford to buy your test and perform it within the available payment, both at present, but maybe even more importantly, as payment evolves over time. And remember, surprisingly, any number of companies actually forget this, the payment for the test has to cover the lab's labor and fixed costs in addition to the cost of your test product. And those other laboratory costs can eat up anywhere from 1/4 to as much as 3/4 of the available reimbursement. So the reimbursement amount that's available either now or in the future does not equal the price you can charge for your test. Now on to those coding and payment specifics that I mentioned. To facilitate access to COVID-19 testing, both CMS and the CPT editors quickly created new coding to describe molecular testing for the coronavirus. The original codes distinguished between tests using the CDC viral detection methodology, coded with U0001, and standard molecular methods for virus detection, coded with U0002 and CPT 87635. These codes didn't initially distinguish among tests on the basis of throughput. They applied to both one-off or standard throughput tests as well as to the initial high throughput tests and platforms. To its credit, however, CMS soon thereafter acknowledged that high throughput tests performed on automated platforms were both valuable to have out there in the clinical marketplace, as well as being more expensive to perform. And so they created additional codes, U0003 and U0004, to describe these high throughput tests. In doing so, CMS defined high throughput as greater than 200 specimens a day and identified several tests and platforms that met this standard. Don't worry, however, if your high throughput platform was not one of the ones initially identified by CMS. CMS has promised to evaluate additional platforms and tests promptly for this designation, and at present, labs can bill using the high throughput codes for testing on any platform meeting the high throughput definition, as long as the labs maintain appropriate supporting documentation about the test performed. So regarding test payment, testing payment was unanimously agreed to by all of the Medicare Administrative Contractors, or MACs, at about $36 for the CDC methodology virus detection tests, and at about $51 for other standard non-CDC methodology virus detection tests. And we presume that the increment for the non-CDC tests acknowledges manufacturers' required investments in methodology development. Subsequently, when CMS established those high throughput codes, they set the payment for high throughput tests at $100, and the same payment rate applies to both U0003 and U0004. This is about 2 to 3 times the amount for standard tests. And this increment provides some lab compensation for the higher cost of the automated platforms and the higher cost of the tests that run on those automated platforms. Turning our attention to serology testing, as testing of activity evolved from just virus detection to also encompass coronavirus antibody detection, in late May, the CPT editors created 2 additional new codes for serology testing. These codes are CPT 86328 for reagent strip tests such as lateral flow assays, and 86769 for multi-step methods such as plate or bead-based ELISA assays. Each of these codes is under a corresponding parent code for non-coronavirus immunoassays that use comparable methodologies. It's also important to note that reagent strip tests are billed on a unit of assay no matter whether a single antibody or multiple antibodies are detected. In contrast, multiple step tests, the ELISA methodologies, for example, are billed per antibody tested, so that there is a difference in the actual billing units for these two types of tests. Now, I mentioned each of these codes is under a corresponding parent code for non-coronavirus immunoassays using comparable methodologies. So CMS followed quickly with payment rates that were established uniformly by all of the MACs. And these payments are about $45 for the reagent strip tests and about $42 for multiple step tests. These codes and payment amounts are fairly new. They're really literally only a couple of weeks old. So we don't yet have a good sense of what commercial payers will do, but we expect them to set comparable payment rates. We also note that the coronavirus serology payment rates are at least 2.5 times greater than the parent code payment rates for methodologically similar tests. So as a test developer, you might be thinking, well, coronavirus serology testing is reimbursing at pretty good rates. I think we can develop and deliver antibody tests under that amount and maybe even leave some revenue for the laboratory that's buying our product. I wonder if that's going to be competitive over the long term. Well, and our perspective on that is that while you can go to market with a relatively high cost and price structure because of the relatively generous initial reimbursement rates, it's advisable that you work to reduce cost of goods as fast as possible. When the public health emergency is declared to be over, and CMS prices these codes on the clinical lab fee schedule, you should expect reimbursement rates to drop until they are at or close to reimbursement for comparable traditional serology tests, or in other words, as much as 2.5 times lower than they are currently. So overall, our key takeaways on the reimbursement and access aspects for COVID-19 testing are the following. First of all, the reimbursement environment for COVID-19 testing is unusually clear for its stage of evolution. Remember, the testing environment is only a couple of months old, but at this point, favorable coverage has already been established. Coding for the various test types and methodologies has been established and appears to be adequate. Payment rates for the various assays, which are effective through the duration of the health emergency, have been set, and various laboratory trade groups have considered that these payment rates are likely to be adequate to cover the cost of testing by the laboratories. So many of the usual uncertainties for reimbursement and access of new tests have been resolved. However, our perspective is that the current environment is about as favorable as it's going to get. And so what that means is that your challenge as test developers is to create COVID-19 tests that not only fit within the currently available reimbursement environment, but also have a cost structure positioned for the long haul. So with that in mind, let me summarize the key takeaways and conclusions from today's webinar. First, biospecimens are the foundation of research. Develop your strategy, deploy your resources, and most importantly, prepare your biospecimen strategy for the COVID-19 market that you'll be entering and the evolution of that market downstream. Second, plan now not only for market entry under an EUA, but for the longer term transition to the 510K environment. Adequate and appropriate planning now will lower your barriers to market entry and improve your competitiveness long term. Finally, when developing your test, ensure that the product's price fits within the test reimbursement, not only for launch today, but for the long haul. And if you do that, you'll be rewarded with a test that remains competitive throughout its product life cycle. And with those thoughts, let me turn it back over to Rebecca to field questions from our audience.
Okay, great. Thanks so much, everyone. Great presentations. I think we're time to move on to Q&A. I want to remind everyone there is still time to submit your questions using that Q&A tab to the left side of your screen. Just before we move on to Q&A, we have two quick polling questions for you. We just want to learn a little bit more about our audience. So let's see, we'd like you to select your company type. So are you IVD medical devices, pharmaceuticals, biotechnology? Are you a clinical research organization or other? And I'll give you all a few minutes to select there. All right. Looks like we've got a nice selection of audience members there. So moving on to our next polling question. What is the biggest challenge you're facing with your COVID-19 R&D? Is it sourcing specimens, applying technology to home use? Are you accessing lab services and logistics, or developing assays that fit within an evolving payment landscape? And again, take some time. This one's a little bit meatier than the last question, so I'll give you all a few minutes to answer that. All right. Looks like, again, we've got a little bit of a wide variety of challenges, which is interesting. So I think now we are ready to move on to Q&A. And again, a reminder, we have a lot of great questions. We're going to try and get to as many as we can within the time that we have. If we don't get to your question, keep in mind we'll try to get back to you individually after the webinar. So first question for the group. So what elements from my EUA submission can be leveraged for 510K submissions? Karen, do you want to kick us off with that question?
Sure. Just to address what I talked a little bit about in the presentation, you know, an EUA has a lot of the same elements that a 510K does in terms of analytical specificity and sensitivity and accuracy. I think one of the areas that you can leverage most successfully is in the design of your analytical specificity studies, because if you, as I mentioned, if you think about using natural versus spiked specimens and really try to make sure that what you're testing for is supported long-term, it may be something to consider early on in the submission, in your preparation process for EUA. I think similarly, if you're thinking about having a test that is either waived or you want for home collection or home test, you know, having those well-designed user studies, some of which have been described already by the agency in templates, some that are pending. But you might also want, when you're doing those user studies, to be thinking more long-term about sample size, study design, maybe making it a little more robust than what the EUA requires, knowing that down the road for the 510K, you might need something more beefy than what is required in the EUA. So those are the kinds of things you want to be thinking about and considering for now and for down the road.
Very good. Next question here. When purchasing specimens from new providers, are there one or two things I should be confirming so I know I'm getting what my team needs? Jim, do you want to try and take this one?
Yeah, sure, I'll jump in. First up, great instinct asking that question. I'd recommend you begin by asking your resources whether or not they are collecting the samples on their own, or whether or not they are procuring them somewhere else and providing them to you. So whether or not it's a COVID program, you need to have full understanding to the accessibility of answers, because ultimately they are going to happen downstream. Examples of answers or questions, that might be asked is whether or not the samples were collected with an IRB approval, or how they were potentially handled. Or if you're really going to get deeper into it, they may also ask you, can you recall a subject and get a second sample? If the source that you're working with is the one that's doing the collection, then obtaining answers to this should be quite easy. However, if there's a middle person or a broker, you need to understand that the path there is not so direct in getting those answers. So as much as possible, understand the method behind your resources and how they're obtaining those specimens. The second thing I'd ask is experience. And this kind of goes back to the slide earlier that I touched upon, the importance of understanding whether or not the site has the right cohort in their practice, and also whether or not they have the right resources to actually conduct your collection. Now, there's really no right or wrong answer to any of these questions. Just as long as you're mindful to the potential impact of the answers and what they may have to the quality of your specimens, then you should be very good.
Very good. Next question, Karen, I think this one might be for you. Is there an expectation from FDA that EUA products are developed under design controls, or is it more of a future-oriented strategy?
Hi, yeah, so this is a future-oriented strategy. So under EUA requirements, there is an exemption for current GMP, including quality system requirements under 820 with respect to design, manufacture, packaging, labeling, storage, and distribution. But it's something to be think about and preparing for, because any of us that have lived through the evolution of the design control process know how challenging and difficult it can be to work backwards. So if you have a system that already is in place and utilizes design controls, this is just a forward and future-oriented way of thinking about a simple transition from EUA to 510K.
Okay, very good. Next question, is there a reference for the need to test HIV-positive samples to validate serology?
So that's a great question, and this is part of the evolution of FDA thinking, panel development, templates, et cetera. So as I mentioned in my webinar, there is an expectation that the assays do not interfere with HIV, and FDA saw some very early data that there were some cross-reactivity. And they had said in the town halls, very specifically, they were looking for 10 of the negatives to be from HIV-positive patients. In looking at the evolution of the templates, so the serology template came out after that particular town hall, and instead of specifying 10 HIV reactive specimens as part of the negative population, they've instead put in that in the analytical specificity section, you need to test for HIV. So that's why I mentioned, so the reference for what testing has to be done will be in the serology template. It does talk about anti-HIV, but I do believe that there's probably two options. One where you're either testing for anti-HIV cross-reactivity in a cross-reactivity study, or that you're testing 10 of the samples from your negative population that are known to be HIV-1 positive. But that piece never got translated specifically into this version of the template, but it certainly has been something that the agency has been talking about. I believe it's also part of the NCI panel. When I was glancing through that, I believe that they do have some samples in that panel from HIV-1 positive patients.
Okay, very good. I think this is another one for you, Karen. Can you comment on the quality system for EUA in contrast to 510K?
Yeah, so this is similar to what I was answering previously, which is you have exemptions under EUA. So as a manufacturer, if you don't have a current quality system in place, you are exempted under the EUA program from having compliance to the 820 part of the regulation. It doesn't relieve you from creating, documenting, and generating and distributing a high-quality product. So it's really important that you understand the principles of properly designing, developing, validating, and distributing a product. But technically, from a quality systems standpoint, other than having, you have to have the ability to report complaints and manage people that are using your product and responding to calls and being able to recall your product if that's something that happens. Beyond those critical requirements, you are exempted under the EUA program from having the full quality system in place.
Good. Next question. COVID-19 R&D is evolving quickly. So when developing an assay, what's the best way to properly test and validate assays while managing full confidence in the data that's generated? Sharon, do you want to take that one, or do we want to? Yeah, there you go.
Sure.
Sorry.
So it is evolving very quickly, and there's some key principles that you can use to think about testing and validating. And number one is I strongly encourage everyone to participate in these town halls, because you get information in those town halls that has not been immediately translated into policy or templates or documents, because we are all asking FDA really good and difficult questions, and they're struggling to keep up with it as hard as they're trying to get this stuff out. You get some really immediate response on that. Secondly, you can look at any of the now EUA authorized tests as a reference for how to test and validate an assay. Beyond what the templates say, you can actually see how this has been translated into actual products that are now on the market. And that's a good way to look at that. And particularly in serology, you have to now be thinking really hard about the fact that, you know, there was this allowance for distribution of tests with the promise to validate. But what we're finding is that that didn't really pan out very well. So there's a pretty limited list of tests now that are actually published, and the data has been reported on for the performance of serology. So those, if you go to the IFUs for those products, you will see what studies have been done to validate. And lastly, if you have something novel, I would suggest that you notify FDA, go through the pre-EUA email process, and ask questions about something that might be novel about how to test and validate your product.
All right, I think we have time for one final question. Let's see, David, do you want to take this one? When it comes to marketing in-home test kits, what are some strategies to be mindful of?
Great question. You know, as I think about in-home testing, to me, I think the biggest thing to be mindful of is that there is no patient cost sharing for COVID-19 testing. And so what that means is, and remember, you're sending out in-home collection kits to be returned. What that means is that you are likely to get a higher than typical rate of non-return of those in-home test kits. Obviously, people want to get their test results. That's why they're asking for testing. But the fact that they don't actually have any patient contribution means that you should factor in, I think, a higher than maybe typical rate of non-return of those test kits. And so you're going to have a little bit more of cost of goods. And remember, you know, you in the end, you can't actually bill for a completed test if you never get the test kit back. So I think that might be maybe one of the most important differences between in-home testing and traditional laboratory testing.
Very good. Well, unfortunately, that's all the time that we have, so that'll be our last question. Again, I want to remind everyone, if we didn't get to your questions, we can try and get back to you individually after the webinar. I want to thank you all for attending this Fierce Biotech webinar. Again, I'd like you to thank you for submitting so many great questions. I want to thank our speakers for participating and Precision for Medicine for presenting today's webinar. The webinar has been recorded. You will be able to access the recording within 24 hours using that same audience link that was sent to you earlier. Thank you again for joining us, and we look forward to seeing you at future events.